Well, hello, young biologists, and welcome to another fascinating virtual lesson. This one covers DNA and the discovery of DNA, why we think that DNA is so important, and who were some of the first scientists to help solving this mystery of what DNA is actually useful for. We've actually known about the existence of DNA for a long time, but nobody really knew what it did or why it was important. The first guy who really did something about that, who really started figuring out things about DNA, was a guy by the name of Frederick Griffith. He was a British scientist, and back in 1928, he conducted some experiments with mice and a certain type of bacteria that causes pneumonia, Streptococcus pneumonia. And one thing he found, there was two different strains of this bacteria. One he called his S strain, and the S strain had a smooth appearance when he looked at it underneath the microscopes. That's what the S stands for. And the other strain looked rough under the microscope, so he called this his R strain. And so the first thing he did, he injected these bacteria live into mice to see what would happen. So experiment number one, he injected the live R strain into the mice. And in the end, he noticed that the mice didn't get sick, they didn't catch this pneumonia, and they didn't die from it. He took the S strain, which is real similar to it, injected it into the mice, and he found at the end that the mice had died. So something was very different about these two strains. There was a different trait, probably caused by a different gene in the S strain than the R strain. His third experiment, he said, was it something in these bacteria that they have to be alive in order to cause a disease? So he heat killed them. He actually cooked them, killed them with heat, and injected the dead S strain into the bacteria. And what he found was that the mice did not get sick and they didn't die from it. So it had to be alive in order to cause the problems. He then did something interesting. He took the heat killed dead S strain, mixed it with the live R strain, both on their own left healthy mice. He mixed these two substances together and injected them into the mice. And what he found was surprising. He found that the mice had actually died just like they did with the live S. When he took samples when he was done, he found live S strain inside of the bacteria. Curious. This was unexpected, and concluded from that that the S didn't come back to life. Nothing can come back to life once it's killed. He was sure they were dead. But what had happened was these live R strain had changed their trait and turned into the S strain. He later hypothesized whatever had caused this transformation from one strain into the other strain must be something called the transforming principle, which he concluded was what genes were. At this time, nobody knew what Mendel's genes were. If you went to look for them in a cell, nobody knew anything about that. But what the first piece of the puzzle Frederick Griffith figured out was that it must be something in this S strain bacteria. Now, coincidentally, he was alive during World War II, and his apartment building was bombed by the Germans, and he was hit by his building was hit by a bomb, and he died in 1942, I believe, before he could really answer the question, what was this transforming principle? What is a gene? And so that question was still kind of left up in the air. Frederick Griffith was not able to answer it. However, a short time later, an American scientist by the name of Oswald Avery took up that challenge, and he set out to figure out what was it in the R or in the S strain that converted the R into this deadly bacteria, and to see if he could see this transforming thing. So what he did is he took an extract from the S bacteria and found it was only this extract that was needed to do the transformation. You didn't need the entire dead S strain. And in this extract, there was really only two things. There was protein and there was DNA. So he ran a series of tests to find out if the transforming material was protein or if it was DNA. He did qualitative tests where he actually looked at it and saw that the material that caused the transformation was DNA. He did chemical tests where he looked at the analysis of what was in the material and found that it had to have been DNA. And then he used enzymes. He actually used an enzyme that got rid of protein, so the only thing that could be left was DNA, and it still caused transformation. He then used an enzyme that got rid of DNA and found that it would not transform after the DNA was gone. And so he concluded that this transforming agent, what caused the change that Griffith had discovered years before, was this molecule DNA. Now again, people rejected this because nobody thought DNA was useful. Nobody knew what it did. And so for a long time, people didn't understand the importance of Oswald Avery's discovery. Kind of like Gregor Mendel before him, he was never really recognized as having made a great contribution to biology. Fortunately for him, years later, two other well-known scientists came and kind of confirmed his results. Their name are Alfred Hershey and Martha Chase, and they were studying viruses, in particular a type of virus called bacteriophage, and that means they're a virus that attacks a bacteria. And the cool thing about them is they're only made of two things. They have a protein coat and they have DNA. And what they do is they transform their host cell, the bacteria cell, into a virus factory. Therefore, whatever they do that transforming with must be a gene because it's transforming just like the other bacteria had done. And so they knew chemically DNA contains phosphorus but no sulfur, and they knew protein contains sulfur but no phosphorus. And so by knowing the chemical makeup, they could use radioactive markers to test whether it was DNA or protein that these viruses were using. And that's exactly what they did. In their experiment, they first marked phages with radioactive sulfur. In their second experiment, they marked phages with radioactive phosphorus. The radioactive sulfur was marking the protein, and the phosphorus was marking the DNA. And so when they did these experiments, they found out that experiment number one, where they marked it with sulfur, the bacteria did not become radioactive. And so this sulfur was not getting injected into the bacteria. In the second experiment, they found that this radioactive phosphorus that was marking the DNA caused the cell to become radioactive. Therefore, it must be the DNA that these bacteriophages are injecting into the bacteria. So likewise, what Oswald Avery had confirmed years before was that DNA was indeed the transforming agent. It is what was causing these cells to change their traits. And therefore, after the time of Hershey and Chase, the early 1950s, we knew that DNA was a very, very important molecule. Real quickly, as a follow-up, just to answer some questions. Who was the first to find the transforming principle? We know that Frederick Griffith in England with his mouse experiment did that. Avery and his team ran 3 tests and found the transforming principle to be, they found it to be DNA based upon the enzymes and things they used. Hershey and Chase ran 2 experiments marking viruses with radioactive phosphorus and sulfur, phosphorus marking the DNA and sulfur marking the proteins. Who confirmed that DNA was a genetic material? Well, the first to discover it was Avery, but who confirmed it was Hershey and Chase, Alfred Hershey and Martha Chase. When a virus attacks a bacteria, the virus injects its DNA into the bacteria, and the bacteria is transformed, is the term we use when it actually changes its trait. Frederick Griffith ran 4 experiments using, he used all of these. He used bacteria that cause pneumonia, he used mice, and he used heat to kill them. So all those things were part of Griffith's experiment. Hershey and Chase performed experiments with bacteriophages and found how viruses inject DNA into their host. That's the correct answer. In Griffith's experiment, he found that the mouse died if it was injected with, now remember the one was heat killed and one was alive, so it was the heat killed S and the living R cells, and these living R cells had changed then into the S and made the mice sick and died. And DNA and protein can be easily distinguished from each other because that the DNA contains phosphorus and the proteins do not. DNA has phosphorus, proteins no phosphorus, but they do have sulfur. Now, although his results were not widely accepted, Avery found that it was the DNA molecule that was responsible for transformation. So there you have it. Fantastic. Hopefully you got a lot out of this lesson and you're ready to work independently on some of your projects. So good luck to you pointers, and we'll see you next time.